Thank you. Yes, okay. Yeah, thanks, Lars, and thanks for the invitation to talk today. I'll be talking about, as you can see, the genomics of healthy aging in eusocial animals. And just to get everyone on the same page, a quick definition or a description of what eusociality is and why I find it interesting. So it's a complex trait, and possibly the most important aspect of this complex trait is the division of reproductive labor among castes. Here we have an example, a cross-section of a termite colony where you have the fertile queen, and then within the same colony you have sterile workers and soldiers. It's important to know that you have these extremely divergent phenotypes, but they all have the same genetic background. So in this case, the sterile workers and soldiers are a direct offspring from this fertile queen and the king. And yeah, the queen and king can also produce the next generation of reproductives. So reproductives and the sterile castes can, in fact, be full siblings. So another fascinating aspect for people working on evolution is that the eusociality has evolved convergently quite a few times in very divergent lineages. So we're going to start off with the least expected one. That's the termites that evolved within the cockroaches around about 170 million years ago. Around about the same time, eusociality also evolved within the Hymenoptera, so basally solitary wasps. And there are several origins in Hymenoptera, several within bees, and then you've got one origin in ants, and then possibly two eusocial origins of eusociality in wasps. But we also have eusociality in beetles. So the ambrosia beetles is at least one species which could be defined as eusocial. And of course, these are weevils, they're called Cleonidae. But also outside of insects, we have eusociality within the rodents, the mole rats. An important aspect to discuss here for my talk and for a lot of my most recent research is the trade-off which we usually see between longevity and fecundity quite often in animals. So you have these short-lived animals like mice that live a couple of years but produce several litters per year, several pups per litter, so like between 30 and 50 pups per year and just live a couple of years, whereas the elephant will live 20, 30 years, but it will only raise a single calf every few years. And there is a trade-off can be seen in experiments in model organisms where it can be manipulated. So you can manipulate the lifespan or the fecundity in Drosophila flies, and you'll see it's usually a trade-off of the other. So it's interesting to note that this apparently does not apply to eusocial animals. So if we have a look at termites again, for example, some termite queens can live for 30 years while laying thousands of eggs every single day throughout their life. So highly fertile, really long-lived, but they show no signs of aging throughout these decades. You can compare that to the non-social sister group, the cockroaches. They just live maybe 100 days. And this is true for all of the origins of eusociality. You have these examples of these really long-lived reproductives, which by far outlive their non-social sister groups, and in a lot of cases show no signs of aging. Let's look at the naked mole rat, for example, which are pretty popular in research at the moment, since they don't seem to contract any aging diseases such as cancer. So with my work, I'm interested in these two aspects: how eusociality has evolved several times, and why always along with longevity, what signatures of that can we find within the genome. So let's talk about some of the results, not only mine, but from the also from other groups, on the molecular signatures involved in these origins of eusociality, and see if we can find any similarities across them. So there are some ideas, some expectations. This is based on a review by Sandra Rehan and Amy Toth, but there are others that support some of these expectations. So lower complex sociality, so let's say subsociality, where you just have parental care, a change from solitary behavior to subsocial behavior, you would expect just some kind of change in behavior. So these are behavioral structures that already exist, but these genes are being expressed in a different context, in different individuals or at a different life stage. So you would expect for the evolution of care behavior, mainly changes in expression. And in the genome, you would expect to find some traces of changes in transcriptional regulation. And as these behaviors become more distinct and you get these casts, these behavioral casts, you would expect to see changes in selection on those different casts, which would be observable in positive selection, protein evolution. And as you get towards these really complex eusocial groups with a highly distinct, rigid, fixed casts, you would expect to see as they diverge more, also the proteins which are important for the different casts to also diverge, and then you would expect to see a greater number of taxonomically restricted genes being important for these casts. And there's quite a lot of evidence in Hymenoptera to support this. So there are a couple of, I'm just looking at 3 comparative studies here which support the increase in transcriptional regulation. There's an example, the figure here we see is from Karen Kappheim's paper from Science in 2015, sorry. So we see with increasing social complexity, you have the solitary bees on the left and the highly eusocial bees on the right, including the honeybee Apis mellifera. And with increasing social complexity, the number of predicted genes which are methylated also increases. So that's a sign for this increased transcriptional regulation, which makes sense if you have to regulate these more complex division of castes. And other results support this as well. Like for instance, in ants, they found a divergence of regulatory elements, transcription factor binding sites, increased numbers of transcription factors. And that all makes sense, right? So, but there's one study here which also nicely shows that with increasing social complexity, you also see greater adaptive changes. So more genes under positive selection. The blue column here on the left, this is the number of gene families under positive selection in the most advanced eusocial lineages. And the simple societies are all the way over here. So that supports, again, the expectations. Another expectation would be, so along with that, you have these expansions of gene families, which also increases with social complexity, at least within this study on bees. And this similar thing has also been found in ants, where they find, so in lots of eusocial lineages in Hymenoptera, you see these expansions of gene families which are related to chemical communication, for example, or development. And that's why we're seeing these olfactory receptors being particularly important and expanded there in the number of copies in the genome. And this is one of the figures showing the increased number of odorant receptors in ants compared to non-social hymenopteran species. And supporting the additional expectation in this Rehan Toth paper, there's also evidence for taxonomically restricted genes becoming more important in ants and in honeybees compared to non-social species. Right, so when I started my postdoc in Münster 8 years ago, they had, we just started this project where we had the first cockroach genome, which we see down here, the brown, written in brown here, the Blattella germanica, that's the German cockroach, and the third termite genome. So with this, we attempted to do the first comparative analysis to see what genomic changes we can see which might be related to eusociality in red lines, in the red branches, and how that compares to evolution of eusociality in the Hymenoptera, which you can see in the blue branches. And indeed, one of the expectations was confirmed, that we found evidence for increased transcriptional regulation here. There's a PCA showing the first three principal components on predicted methylation. So we see the distribution of methylation across all genes. And down here, grouped on the left, we have all the termites, the solitary cockroach, much further away. And it's important to note that the methylation patterns here clustered very strongly within the termites and very far away from all of the other species we looked at, indicating a strong change in methylation patterns in the root of termites, which remained conserved throughout the evolution of termites. And this could be related to caste-biased expression. Furthermore, we found significant changes in zinc finger transcription factors, so obviously important for transcriptional regulation, and these are enriched for queen-biased genes, so genes which are important for the regulation of queens compared to workers. So probably important for the evolution of castes in termites. Another expectation we had was that the odorant receptors, which were highly abundant in, or are highly abundant in Hymenoptera, especially in eusocial Hymenoptera, these would also be important in termites and expanded in termites compared to non-social outgroups. At the bottom here, we see the structure of the odorant receptor in yellow. And surprisingly, odorant receptors don't seem to be important, or as important in Blattodea. In fact, they're not very abundant at all. In contrast, the inotropic receptors, which are a completely unrelated group of chemoreceptors, as you can see from the structure here, it's very different. These are highly abundant in cockroaches and termites, and probably important for caste-specific roles, for example, communication, colony communication, which is evidenced by castes. So a lot of these copies of inotropic receptors had caste-biased expression, so important for possibly different roles in different castes. Another surprising aspect was that we hardly saw any gene family expansions. So this is what had been talked about a lot in Hymenoptera with the evolution of eusociality. So great novelty, these different castes evolving. There were massive expansions in certain gene families, and we saw more contractions than anything else. This surprised us slightly. So at the bottom, the fourth column from the bottom, fourth bar at the bottom, this is the number of proteins in the cockroach, and the bottom three, these are the number of proteins in the termites. So quite a reduction. And the same goes for the genome. The genomes are smaller in the termites. And we have more genomes since then. So this was published 2018. There have been more genomes since then. And we see in this right-hand bar plot, the genome size in the published or the available cockroaches are significantly larger, much larger than in the termites. So if we were only looking at termites, we might conclude from this that eusociality leads to a reduction in genome size and proteome size. But we already know from Hymenoptera that that's not necessarily the case, where the opposite seems to happen. Genomes tend to be slightly larger, albeit not significantly. You see all of these expansions in the proteomes. So on the basis of it, a lot of the changes that we see seem to be lineage specific. And if we have a look at, this is new data, we've recently sequenced wood roaches in the yellow bars here, which are the sister group to all termites, and they are also wood feeding and perform parental care, so they're subsocial, but not eusocial, they don't have castes. And you see here that the genome size reduction already probably occurred at the base of wood roaches and termites, so it's more likely linked to the ecological specialization of wood than anything to do with eusociality. So we see here, I mean, it makes sense, the more genomes we have, the more information we have, so we have to be careful with any conclusions if we just have a limited number of genomes. This is a work in progress. We have more genomes, and then soon we'll be able to say more about this. So I talked about the reduction in proteome size, so lots of contractions And on this branch here, we see there were 10 significant contractions at the root of termites and only 7 expansions, whereas there were lots of expansions in the cockroach. But we saw TE's transposable elements were likely important for the expansions of gene families which so the gene families which did expand significantly in the termites compared to outgroups, it was likely influenced by TEs in flanking regions. So we saw a significant enrichment of TEs in these gene families compared to those that were expanded, for example, in the cockroach. So we have TE activity being important for novelty here. More recently, we've been looking for other mechanisms for novelty, so the novelty of eusociality in termites, since we see reduction in the proteome. And we find, although we have fewer protein coding genes in the termites, there have been some major domain rearrangements. In the red square at the root of all termites, and in the blue square here, this is at the root of true workers, where workers were truly sterile. And we have a lot of domain rearrangements occurring here, which could be important for the evolution of novelty in the termites. So just to summarize, if we look at the different origins of eusociality that have been looked at in genome so far, we seem to have a consensus that increasing transcriptional regulation appears to be important, and the adaptive evolution of chemoreceptors. However, there are lots of differences. So odorant receptors in Hymenoptera seem to be important, whereas inotropic receptors more important in Blattodea. So we have the same selective pressure there for chemical communication to evolve, but you have different origins and different molecules which are being selected upon. TE seem to be important for the evolution of novelty in termites, whereas they appear to have a reduced importance in eusocial Hymenoptera. We have a proteome and genome reduction in termites versus expansions in eusocial Hymenoptera. Domain rearrangements are probably important in termites, for these, as far as I know, have not been studied yet in Hymenoptera. Yeah, and to continue on trying to address this question, we just need more genomic data, and that's something we're working on at the moment. We've also been trying to understand the molecular mechanisms connected to the healthy aging in eusocial organisms, why there appears to be a decoupling of this longevity fecundity trade-off. So in Cardiocondyla, in ant queens, we had some transcriptomic data for old and young ant queens, and I constructed a co-expression network, which we can see here. And in this network, each of the dots, each of the dots represent genes and their connections. And the edges in this network represent the correlations of the expressions, of the strength of the co-expression. So the closer they are together, the more strongly co-expressed they are. And the colors represent different modules within this network, which are particularly strongly co-expressed. And we see on the right, in this, we have several modules, my cursor's not working, several modules which are clustered together that are upregulated in old queens, and then on the left you have this cluster of modules which are upregulated in young queens. So these parts of the network are activated in young or in old queens. And we had a look at which genes belong to these modules which are up or down regulated. And in the old modules, and the modules that are upregulated in old queens, we find lots of functions which are important for combating classic hallmarks of aging, such as related to maintenance of proteostasis, transcriptional regulation, and stress response, among others. Another thing I wanted to look at was the selection pressure, or the selection efficiency on genes which are expressed at old age in ant queens. So one of the evolutionary theories of aging is the mutation accumulation theory, where you have this selection shadow at old age, meaning, so this is the survival curve here, the black line is a survival curve for populations in the wild. Just through extrinsic mortality, you have fewer individuals at increasing age. So the effective population size decreases with each age group. And so the selection efficiency also decreases with each age group. So any gene which is important for survival at old age would not be efficiently selected upon, especially compared to genes which are important for survival at young age. So that's the brief description of what the selection shadow means. And the red line here shows a typical survival curve in captivity or in protected conditions. So I wanted to test whether we would find a selection shadow in the ant queens. And first of all, I want to do this with DNDS. So just to briefly explain what DNDS is, that relates the non-synonymous substitution rate to the synonymous substitution rate in coding regions. And if the non-synonymous rate is equal to this synonymous substitution rate, is an indication of neutral evolution, so close to the mutation rate, the neutral mutation rate. And anything closer to zero, so a smaller dn is compared to ds, we have purifying selection, and anything greater than 1, positive selection. So in the case of a selection shadow, you would expect values to drift. So purifying selection, we would expect to dominate. So we have DNS values between 0 and 1 in most cases. So in the case of reduced selection due to a selection shadow, you would expect values to be closer to 1. So greater than 0. And this is what we see for Drosophila. So in blue, we see the DNDS values for genes which are expressed, which have high expression in young females, and in red, the genes, DNDS for the genes which are expressed in older individuals. So which ones that you probably include genes which are important for survival or for fitness at older age. And these do have an increased DNDS values indicating weaker selection. We did a couple of other analyses which corroborated this. So DNDS is capable of detecting a selection shadow. And something similar was also published for humans. And did the same in the ant data, and we didn't see this. In fact, we saw the opposite. So there's no sign, at least with DNDS, we found no sign of a selection shadow. So genes which are expressed in these older queens are actually under stronger purifying selection. In a further study, we did a co-expression network analysis for termites. So obviously completely unrelated to the ants. These are Latodea, so social cockroaches. And again, we found parts of the network which were activated in young queens, other parts which were activated in older queens. And when I compared these networks to each other, I found no similarity in the young queen modules, but there was a lot of overlap in the older modules. So these modules which are upregulated in the old queens, they had similar structures. So there was a high preservation statistic, indicating convergent expression of structures within these networks, and also shared functions, mainly due to protein transport. So another thing we can do with co-expression networks is make some assumptions about transcriptional regulation. So this is something that had been done before in mouse brains. They did a co-expression network for young mouse brains, for old mouse brains, and they found that the connectivity, so the strength of the correlation between the nodes, between the genes, was lower, much lower in the old brains. And they saw this as an indication of a breakdown in transcriptional regulation. That's one of the major hallmarks of aging. And that makes sense. And so I checked to see whether we would see this in the ant and the termite networks. We didn't. In fact, we saw a slight increase in transcriptional regulation. So we see no decline in connectivity. So it appears that the genes are still really well regulated in these old queens. But then looking at a more specific pathway rather than the whole network, if we look at the nutrient sensing pathway, so IIS, so insulin signaling, and the TOR pathway, this is quite central to the trade-off between longevity and fecundity. So an upregulation of this pathway through a high glucose diet, for example, would be advantageous for reproduction, but would shorten lifespan, for example. So I had a look at the correlations between all of the gene members of these pathways. And one thing was striking, which was different between the ant and the fly, and that was the correlation between RHEP, which is the positive regulator of TOR, the central player in the TOR pathway, but it was negative in the ants. I mean, it's only RNA now, I know, so it doesn't mean that it necessarily inhibits TOR, but it's a first indication that something may have changed in the pathway here. And interestingly, we find the same negative correlation in the termite as well. This is something we still need to look into to see whether we see the same sort of relationship on a protein level. And we're doing some first RNAi experiments, which we would like to expand and also carry out in termites. But it would be great if we could find some kind of rewiring going on in this pathway, because that would help us understand why this trade-off doesn't seem to occur. So the insulin-like peptides are also important in this pathway. So with the glucose, with the intake of glucose, the expression of insulin-like peptides increases, and that activates this pathway. And it's known that increased ILP, so insulin-like peptide expression, reduces lifespan. And in the termite, we found one ILP, which we call ILP9, which had a massive increase in expression along the lifespan. So comparing young founding queens to 3 month, 9 month, 31 month, and the old queens, which were over 20 years old, we saw a massive increase in expression. And this coincided with high glucose levels, which were found in the hemolymph. What's startling about this is that this would normally have negative consequences due to fat storage, which doesn't seem to be happening here. In fact, the glucose, it appears that the glucose is being transformed and being converted, or the energy from that is being converted directly and sent straight into reproduction rather than fat storage. This is something we're trying to understand better with some RNAi experiments on cell cultures. We also had a look at methylation, how important methylation is for the regulation of Cas-biased genes. And for this experiment, we had 4 different phenotypes. We had the female workers, which are short-lived and sterile. We had 2 different aged groups of queens. So we had the virgin queens, so they're young and fertile, and then the mature queens, which are over 20 years old, highly fertile, and also mature kings, which are also highly fertile and over 20 years old. And we found, so as in most insects, there's a positive correlation between expression and methylation, but this differs depending on which region of the gene you look at least in these termites, and also whether a gene is differentially expressed. So here we have on the x-axis the expression, and on the y-axis the methylation level. And we see if we look at the exons and the 3' flanks, so downstream of the gene, we see this positive correlation of expression and methylation, but this is much stronger in genes which are caste-biased, so differentially expressed either in the different castes, so the individuals which have different levels of fertility and longevity, or at different ages, so also differ between the young queens and the old queens. So some kind of regulation of these genes is going on with methylation, especially in the 3' flanks, where there's no correlation between expression and methylation in the non-biased genes. And something similar is happening for genes which appear to be alternatively spliced in the different groups. Again, especially in the 3' regions, we see this strong positive correlation between expression of methylation, which we don't see in the genes which are not alternatively spliced. Another mechanism we believe is important for survival in the queens is that they manage to defend much better against TE activity and the invasion of transposable elements into the genome, which cause an accumulation of, normally without protection, cause an accumulation of mutations in the genome. But we see here the proportions of repeat expression within all of these different castes and age groups of the queen, and we see, especially in the old queens, very low expression of TEs. And this is due to an increased TE regulation by the piRNA pathway. This is similar to the differences in transposable element regulation that you see in non-social individuals in the germline compared to the soma. So just to summarize what we have there, some of the mechanisms, how they're managing to live so long, well, there appears to be really well-regulated transcription, methylation, also transposable element suppression in eusocial queens. We find convergent expression patterns in a termite and an ant, strong selection on old genes, possibly due to extrinsic mortality, but a lot of people would disagree with this. Possible switches in the nutrient sensing pathways, and this is something we need to look into with lab experiments. And I've been thinking about this a bit more. Can we really expect convergent molecular signatures across all eusocial origins within the genomes? And if we think about how So with increasing social complexity, you have a decrease in effective population size because you have these larger colonies and the more clearly distinct castes, so very absolutely sterile workers. So the relative number of reproducing individuals decreases. So with increasing social complexity, you have decreasing effective population size, and this should lead to a relaxation of selection in social lineages, which correlates with social complexity. And this is something you should be able to see in the genomes, right? You can see the effect of selection in coding regions. So I tested this in Blattodea, in the termites and cockroaches, and again used DNDS as a proxy for relaxed selection. So as we, from bottom to top, we see increasing social complexity. All the ones with green dots are termites, and the ones at the top actually have truly sterile workers that can never reproduce. And we also have increasing lifespan with increasing social complexity. But confirming our expectation, we also see higher DNDS values in the termites, and especially in those that have sterile workers. So this would at least confirm our expectation of relaxed selection. And if we have a look in mole rats, some genomes, 2 mole rat genomes exist, and I did DNDS analyses for these as well. So we have the naked mole rat, NMR, and the Damaraland mole rat, which is also socially eusocial, but possibly less socially complex. And then some other rodents which are communal. We see the highest DNDS values in the mole rats, which correlates again with social complexity. And further analyses confirm that these high DNDS values were due to relaxed selection, especially on the branch where sterile workers evolved in the termites, and on the branch leading to the naked mole rats, we have relaxed selection. And similar results have been presented for Hymenoptera. So there we have it. We have clear signals of eusociality in the genome. It correlates with social complexity and in multiple eusocial lineages, in 3 independent lineages. We don't only have relaxation in termites. Of course, there's also intensification there, and positive selection because we have novelty evolving here. So there were also some genes under intensified selection where the sterile workers evolved. And these genes, under increased selection, were enriched for old bias genes, so genes which are important for fitness, survival, and reproduction in the old queens. So a higher proportion of those were overexpressed in the older queens compared to young queens and female workers. And similar things have been found in the mole rat, so positive selection on lifespan genes there. And I presented earlier in this presentation that the strong selection of old queen genes in ants as well. So there again, we see this selection for longevity in 3 independent lineages. We can also expect signatures of selection based due to differences in selection on the different castes. So the queen has sole responsibility in these really highly complex, socially complex species. Their sole responsibility is reproduction, and their effect on the fitness of the colony is direct, of course, and they're also well protected. So this is quite different to the selection regime on workers and soldiers, which are exposed to many more different kinds of selection pressures. However, their selection is indirect, so their effect on the colony fitness is lower, just because there are fewer individuals, and it's also, more individuals, so the effect of each individual is lower, and it's also indirect. But nevertheless, you would expect to see very different selection patterns on genes which are associated with the different castes. So if we have a look at that in the genome, cartoon genome here, so we have 3 different mock genomes, 2 eusocial and 1 non-social genome. And the different colors represent whether a gene is expressed in all individuals equally, or just in queens and just in workers. In reality, it's never, or very seldom, only queens and only workers, but this just as an example. Let's say the orange ones, worker genes they're important for worker survival, and the green ones are important for queen survival. You would expect to see a divergence in selection patterns between the orange and green genes within one genome, because they're important for very different phenotypes which have different selection regimes. But you might see a convergence in the selection patterns on orange genes in 2 completely unrelated lineages. So selection on workers in ants, you would expect to be possibly more similar to selection on workers in termites than on queens in the same species. And again, getting onto the transcriptional regulation, which has already been shown in genomes, we already have multicellular organisms here. So we already have complex transcriptional regulation, right? Because genes need to be regulated, turned on and turned off in different tissues and different developmental stages. But we have an extra level of complexity here because that all needs to be done in different individuals. So you would see an additional layer of transcriptional regulation. So these 2 aspects together, the regulation and the different expression, should lead to some very complex patterns, which you may expect to somehow converge, be more similar between eusocial lineages than between eusocial and non-eusocial. And these are really complex patterns, I would expect, and it's something I think we may be able to find with machine learning techniques, and it's something I've already tested just to see whether it's feasible. And I developed a tool which classifies sequences into originating from a eusocial or non-social genome. And I tested this on 10 available eusocial insect genomes and 10 non-social insect genomes. This was because at the time we only had 5 termite genomes. So 5 termite genomes and then 5 eusocial hymenopteran genomes. And then to balance it all out, I took 10 non-social genomes from different orders. And from these genomes, I extracted different features from throughout the genome. So the genomes were split into exons, each intron separately, each 5' flanking region, 3' flanking region, and everything else in between was called intergenic. And from each of these individual sequences, I extracted hexamer scores to represent the sequence content and motifs within the GC content as well. And I trained the algorithm on a subset of that data to identify patterns which differentiate eusocial and non-eusocial genomes, and then tested on the rest of the data. So it took a sequence in the end and then decided whether it was eusocial or non-eusocial, so a binary classification. And the classification accuracy was really high, yeah, especially in non-coding regions. So in the flanking regions, we would expect these regulatory mechanisms to be present. It was a high accuracy. It managed to determine whether the sequence came from eusocial or non-eusocial genome around about 80% of the time. Whereas the exons were not informative at all. And I believe this is sort of a nice control showing that it's not just detecting phylogeny, it's not detecting or somehow managing to identify a termite genome, because then you would expect the signal to be stronger in the exons. So this was a proof of principle, and it's nice that it seems to be able to do this, and this is something we're working on. We want to expand when we have more data. I then wanted to see whether it would be helpful in determining whether a gene is more important for young age, old age, or longer lived and shorter lived individuals. So I labeled genes with their expression bias in terms of whether it's more strongly expressed in a worker or in a queen. So the long-lived queens, the short-lived sterile workers. So instead of a binary classification, now we have more of a kind of a regression. You have a value for each gene, which shows how biased it is towards a certain caste in terms of its expression. But I use the same features and split the gene body into the same regions. So the 5' flanking region, 3' exons and introns, and then it should predict the expression bias. I did this on 2 data sets, on the termite data set and also a published data set for naked mole rats. I measured the median absolute error, which is basically just looking at the absolute difference between the true value and the predicted value. So if it was working perfectly, you would expect the median absolute error to be 0. The values were in fact greater than 0, but still relatively close to 0. And if we just took random values from so we just took the true values and randomized them, we would be getting error rates of which are much higher. So yeah, I mean, it seems to be doing something. It's not working perfectly. It's still not dealing very well with the extreme values, but it's at least it's a lot lower than you would expect by chance. And we've done the same with genes which are differentially expressed throughout the lifetime of queens, so young age and old age, to see if we can find anything in the genome which is important for the regulation of these genes. And we also find quite low error rates. So that's all I have to present today. I'd just like to summarize what I'm working on. So I'm interested in how eusociality evolved in diverse lineages. It's important that queens are highly fertile and long-lived, apparently in all eusocial lineages. So I'm interested in the molecular signatures in these multiple origins. We've talked about the decoupling of the longevity fecundity trade-off. But my goal is to try and understand the molecular mechanisms of healthy aging, also in non-eusocial animals. So that's the direction I'm moving in the future. I'd like to thank all of these people and many, many more who have worked together with me on this, and I'd like to thank you for your attention. I'll be happy to take any questions.